Hello and welcome to our first biochemistry lecture in Bio 120. Today we're going to be looking at nitrogen assimilation and the nitrogen cycle. So this image shows the way in which you have the oxidation and reduction of nitrogen on Earth. We're going to be looking at how gaseous nitrogen is being brought inside the biological systems, how that nitrogen now in the form of ammonia through oxic reactions will become nitrite, and then later how that nitrate is going to now be converted back into nitrogen gas through the process of denitrification. In here what we're having is a table showing the process keys and the microorganisms that are going to help us understand the nitrogen cycle. We're going to be looking at nitrification reactions in which ammonia is going to eventually be converted into nitrite. We're going to see that there's two different kinds of microorganisms that work together to bring these two reactions. We're going to look also at the process of denitrification, which is made by many different organisms, including Pseudomonas. And now we're going to also look at the fixation of nitrogen, in which nitrogen gas from the environment will be brought into the biological system. And this can happen both by free living organisms or organisms that are in symbiotic relationship with plants. Last but not least, I would like to introduce you to the Anammox reactions, which are going to be mediated by Brocadia. So when we have the decomposition of biological matter from animal, plants, or bacteria, you're going to have the production of free ammonia. So you're going to have those tissues that are going to be degraded into primarily protein polymers. Those protein polymers eventually is going to be broken down into smaller peptides and amino acids. Those will then be translocated across the bacterial membrane. And inside the bacteria, in the cytoplasm, the molecules, especially the amino acids, will then be deaminated. For example, you can have lysine be deaminated into acetate and ammonia. And we're going to see that in that reaction now, the microorganism could use that acetate, for example, as part of the acetyl-CoA cycle, and the ammonia will then be used to bring nitrogen into the biological system. So what's happening to the ammonia that is produced from the degradation of plant or animal matter in the environment? Some of them is going to enter the atmosphere. Other ones is going to enter the soil layer in the geological cycle of nitrogen. Some of them is going to enter the stream and rivers and lakes, and that could be one of the main sources of algal blooms that we see in some of the rivers and lakes that we have around here in Merced County. And eventually, that nitrogen will be acquired for cell growth and recycled biologically, or completely enter the geological cycle, which is non-biological, as you know. So the first reaction that we're going to take a look is going to be the process of nitrification. Now, nitrification is going to occur, as I showed you earlier, in two different steps. We're going to have the oxidation of ammonia into nitrite, and that's going to happen in one set of organisms. The other set of organisms is going to take the nitrite from the previous organism and use it and oxidize it to form nitrate. These two processes are going to be respiratory, and they're going to use electron transport chains, and oxygen will be the ultimate electron acceptor. That's why they're going to be considered oxygenic and respiratory. So they have to occur under aerobic conditions. Now, depending on which reaction you're looking at, you're going to either have ammonium or nitrite as an electron donor. The electron acceptor is always going to be oxygen, and you're going to result by the electron transport chain of these reactions into a proton gradient, and that proton gradient will be used to produce ATP. Now, as a source of carbon, many of these microorganisms are going to fix carbon through the Calvin cycle, just like plant cells do. In here in this table 13.1, what I am showing you are the energy yields from the oxidation of some of these compounds. So the first one is going to look at the oxidation of ammonium into nitrite by nitrifying bacteria. As you can see from this table, that is going to produce about minus 274 kilojoules per reaction. And Now the oxidation of nitrite into nitrate, this R over here that you see in the middle is the reaction. That's going to be also worked by other types of nitrifying bacteria, and that is going to have a delta G naught of minus 74.1 kilojoules. So both of these reactions are going to be energetically favorable for the microorganism. The first set of reactions are going to be carried out by some of the species of nitrosomas. And nitrosomas are marine gram-negative cocci. They're going to use ammonia during oxidation and convert it into nitrite for energy. The electron donor is going to be ammonia. The electron acceptor is going to be oxygen. And the reaction, I am showing it here at the bottom. Here is the ammonia. Over here is the ammonium ion, but here is ammonia. And it's going to take 1 molecule of ammonia and 1.5 oxygen molecules and give you a nitrite plus a water molecule. Since these reactions are happening oxygenic, they are going to involve electron transport. And notice inside the microorganism you have a very dense array of membrane which are going to be required for increased surface area to the reactions to happen. So this is an obligate chemo-lethal autotrophic microorganism. Now, as I mentioned, that's why they have the internal membrane array that is shown in this electron micrograph of Nitrosococcus oceani. So the membranes, as I mentioned, are designed to help increase the surface area where the reactions for energy are going to be occurring. So this image over here from figure 13.26, it's showing you the steps in which the electron transport chain for the oxidation of ammonia to nitrite are going to happen. The first reaction that I want to point out to you, it's the reaction in which the enzyme ammonia oxidoreductase, it's going to take ammonia and oxidize it into hydroxylamine. So the enzyme ammonia oxidoreduction, or AMO, it's going to take ammonia and oxygen and protons, and that is going to give us a molecule of hydroxylamine and a molecule of water. That molecule can diffuse freely across the membrane, and in the outside you're going to have the second step, which is going to be the oxidation of hydroxylamine. The oxidation of hydroxylamine, it's going to now take a hydroxylamine molecule and a water molecule, eventually providing you a nitrite, and 5 protons. Here are the protons being used that are going to now help with the they're going to help with the proton modal force. So now the hydroxylamine oxidoreductase, or HAO, which is going to catalyze the last part of this reaction, is going to eventually pass electrons to cytochrome C. And cytochrome C, usually passing 2 electrons at a time, is going to eventually bring the electrons down to a second enzyme, which is going to be the next part that I'm going to show you now. So here, what we're going to have is the actual reduction of oxygen by the cytochrome AA3 complex. So the electrons that have come from the initial reaction are now going to be delivered by cytochrome C, which is, as you know, a mobile electron carrier, and bring it down to cytochrome AA3. And in here is when you have the reduction of oxygen. Oxygen molecule, for some of the protons being generated, are going to generate a molecule of water, and that's going to also contribute to the proton modal force. At the end, 3 protons are going to go across the ATP synthase to generate 1 ATP molecule. As you are aware from Bio 110, usually you require, and for an entire turn of the ATP synthase, you require 9 protons going across to produce 3 ATP molecules. So this is a respiratory reaction where the initial electron donor is going to be ammonia and the final electron acceptor is going to be oxygen. This reaction is going to then produce nitrite, and nitrite is eventually going to diffuse outside of the cell into the marine environment. This is where the second microorganism comes into play. Here we're going to look at some of the Nitrobacter species, and here we have Nitrobacter winogradsky. And Nitrobacter winogradsky is also a marine gram-negative bacillus, so it's an aquatic microorganism. Notice as well that it has an extensive membrane structure inside its cytoplasm. So this organism is going to receive electrons donated from nitrite, and the reaction is going to eventually pass those electrons to oxygen. So the initial electron donor is going to be nitrite, and the electron acceptor is going to be oxygen. The reaction is shown down here at the bottom, where you have a nitrite molecule and an oxygen, half an oxygen molecule, and that is going to give you a nitrate molecule. As previously, this is a chemolethal autotrophic microorganism that is also going to use the Calvin cycle to fix carbon dioxide. And as you can see, it has an extensive membrane array inside the cell. Here in this other image, you're now having the electron transfer system that is going to contribute to the generation of energy in this organism. So we can begin the reaction by the oxidation of nitrite into nitrate. So a nitrite molecule is going to donate its electrons, and it's going to take also a water molecule to now make a nitrate. The electrons will be passed to the enzyme complex nitrite oxidoreductase, which also is going to pass them down to a cytochrome C molecule. Those electrons eventually are going to reach its final destination, which is going to be a cytochrome AA3 complex, and in that cytochrome AA3 complex you have the reduction of oxygen into water. This reaction is going to now allow protons to be moving across the membrane, and those protons moving across the membrane will again be used to produce ATP. So both these microorganisms are going to be chemolethal autotrophs. This is one of the first examples that you have learned about the biochemistry of an organism that is a chemolethal autotrophs. So they're going to acquire their energy from an inorganic compound, either it's going to be ammonia or nitrite. They are obligate aerobes, meaning that in order for these reactions to happen, they're going to use oxygen as the ultimate electron acceptor. And as shown in the previous graph, here is the delta G prime naught of the reactions. So here we have ammonia oxidation to nitrite producing minus 275 kilojoule per mole of energy, and nitrite to oxidation to nitrate producing minus 76 kilojoules per mole. These organisms cannot really use an organic source of carbon for growth, and both of them must fix carbon by the Calvin cycle through acquisition of CO2. So now that you have produced nitrate, the nitrate then accumulates geologically, unless an organism is going to use it. So we can then later talk about the fact that there is going to be a lot of contamination of nitrates that can be detrimental to the environment. So let's look at the second set of reactions that are going to be looking at. Actually, let me just put them together for you in one more way. This is from a article that Dr. Martin Klotz from the University of Louisiana in Louisville, Kentucky produced. Here we have the cooperation of nitrification from both bacteria, where they take ammonia, converted into hydroxylamine by the reaction that we discussed earlier. Hydroxylamine later donates electrons, and eventually you get nitrite. Nitrite is later going to now be completely oxidized into nitrate, and nitrate, for example, can go down into the ocean floor. What we're going to look later, it's the arm here on the right, where we're going to look at the denitrification, which is the process in which now nitrate, it's going to be converted into nitrogen gas. One other reaction that I want to discuss before, it's going to be the Anammox reaction. And the Anammox reaction is shown here in the blue box. It is a reaction that is happening anaerobically. So it is happening in the absence of oxygen, and it's going to take some of the nitrate in order for it to now convert it into nitrogen gas. So now the Anammox reactions is going to help with the oxidation of ammonia under anoxic conditions. We're going to be looking at Brocadia anammoxidans, which is the microorganism that has been shown to produce this kind of reaction. They are gram-negative bacteria, and they belong to the phyla of plantomycetes. The plantomycetes of bacteria doesn't have a peptidoglycan layer. They are anaerobic chemolithotrophic microorganisms. The amazing thing about them is that they're going to be generating their energy for cell division, and they have a generation time G between 10 to 30 days. So the process by which they're going to generate energy, it's efficient to allow them to grow, but it takes time for them to actually undergo cell division. What I want to show you here is this picture on the left. Here you have the Brocadia anammoxidans, and they are cocci. Here you have one microorganism with the transmitted electron microscope image, and what I want to show you is this very large internal organelle that the microorganism have, and that is going to be called the anammoxosome. So under anaerobic conditions, this microorganism is going to be able to oxidize ammonia and produce nitrogen gas. That reaction, it's going to be happening in the anammoxosome. Now the anammoxosome, it's an interesting organelle, and the microorganism itself is actually quite interesting. It doesn't have peptidoglycan, so it has a cell wall, shown over here in the left, that is completely made out of protein. After that, it has a cytoplasmic membrane right underneath the cell wall. But then this microorganism has another intracytoplasmic membrane. So it has a double membrane system. In between the cytoplasmic membrane and the intracytoplasmic membrane, you have a region called the periphoplasm, shown over here. Now, the riboplasm is what we consider to be the cytoplasm, called riboplasm because you have ribosomes in that area, and the nucleoid of the bacteria is contained within the riboplasm. The anammoxosome, it's now this very large organelle found inside the microorganism. It has membranes, and it has its own membrane surrounding the organelle. What is interesting about this membrane is that the lipid bilayer contains a special kind of fatty acids called lateranes. And the lateranes are called such because it contains these cyclobutane rings that looks like a ladder, and those kind of fatty acids are now joined together to blue glycerol molecules shown over here. Well, the molecules are not blue per se, but in the image they are. Now the laterine lipids, because of the way in which they can stack, are able then to increase the impermeability of the membrane to protons and a molecule called hydrazine, which we're going to look at in a moment. This microorganism is a chemolithoautotroph and is going to combine ammonia with nitrite as the electron acceptor, and eventually that is going to take nitrite and produce nitrogen gas using a hydrazine, which is an N2H4 intermediate, and hydroxylamine as an H2OH intermediate. Now, this reaction is shown over here into the right, and we're going to look at 3 different enzymes. Hydrazine hydrolase is the enzyme HH. You have hydrazine oxidating enzyme, which is the enzyme HZO, and you have the enzyme nitrite reducing enzyme, which is NIR, NIR shown here. The reactions that are happening anoxygenically for this bacteria are as follows. An ammonium molecule is going to be combined with the hydroxylamine molecule by the enzyme hydrazine hydrolase, producing water and a hydrazine. The hydrazine is here now shown in the internal portion of the anammoxosome. Now, hydrazine, it's now going to donate its electrons to the enzyme HZO, and by a series of reactions, you're going to produce 4 protons and a nitrogen gas molecule. 4 electrons are going to be also then donated. Those 4 electrons eventually will combine with 5 protons as well as a nitrite molecule, and that reaction will happen in the nitrate reducing enzyme, which is going to produce a water molecule and hydroxylamine that can then be used again to combine with another ammonia. The protons generated when the hydrazine donates its electrons are able to pass through the ATP synthase in the membrane of the anammoxosome and produce ATP. I wanted to take a look at this image because the one in our book, it's actually incorrect, and it has the direction of the production differently. So I went to the original paper who produced made the discovery, got the diagram that they use. So make sure that you can compare it to the image that we have in our textbooks and see where the image in the textbook is wrong. In order for this organism then to be able to take an ammonia molecule, here's now showing an ammonium ion with a nitrite, and eventually give you a nitrogen gas and water, that is going to have a delta G of minus 357 kilojoules. That reaction needs to be performed 15 times in order for the cell to gather enough energy to be able to fix one CO2 molecule. And that fixation is going to happen by having a CO2 molecule and take some of the nitrite made by the cell and eventually form this nitrate that is going to be released. So it's very energetically demanding to fix carbon, and therefore that reaction for ammonia oxidation has to happen close to 15 times to produce enough energy to get the reaction of the carbon fixation going. So now you have looked about two different ways in which ammonia can now be converted into nitrite. One of them oxygenic and the other one anexogenic. Now let's take another look at a different reaction, and this is going to be the reaction of assimilation. Assimilation, we're going to take a nitrate, that is a nitrate, that is going to be eventually brought back inside the biological system as a form of an amine. So to assimilate the nitrogen from nitrate, the cell has to perform the assimilatory nitrogen reactions. And this is very simple. You're going to take the nitrate, actually it should have a minus at the end, 8 protons, and that is going to produce an ammonium ion, and that ammonium ion eventually can be put into protein. These reactions are not energy producing reactions, and it is catalyzed usually by enzymes in the cytoplasm, and most organisms have them. So bacteria, archaea, plants and fungi are able to catalyze those reactions. So it's a very direct way in which you can bring nitrate from the environment into ammonia. We're going to look that this is one of the ways in which hydroponic reactions work. Now, let's take a look at the other spectrum, and this is going to be the denitrification reactions that are going to happen in the absence of oxygen. And in this case, we're going to take nitrate and bring it back into nitrite by denitrification and convert it into nitrogen gas, either directly or through an intermediate of nitric oxide or nitrous oxide. Now, these nitrate reduction reactions are going to come in two different ways. As I mentioned, nitrate reduced completely to nitrogen gas, or nitrate eventually reduced to ammonia. Both of them are going to happen. They are respiratory process for energy harvesting, but they are not oxygenic respiration. They are only occurring under anaerobic conditions. So the enzyme eventually that are going to perform these reactions, they're membrane bound, and the nitrate are going to serve as the electron donor, and they are either going to make formate, which is shown over here to the right, or they are going to generate NADH that the cell can later use. They come in two different flavors. Dissimilatory reaction are reactions in which the product is going to be released into the environment, or an assimilatory reaction in which the product is now going to be assimilated in biological molecules within the cell. So let's look at some of the nitrate reducing bacteria. We're going to look at the denitrifiers, where nitrate is going to be produced to nitrogen gas. We have also the nitrate ammonifiers, which are going to take nitrate and make it into ammonia. Nitrate reduction to ammonia yield a little bit more energy than nitrate reduction to nitrogen gas, and the cells are going to have a different delta G. Here are part of those reactions, and we have two different organisms that can perform these reactions. We're going to start with nitrate, NO3 minus, and nitrate is going to be converted into nitrite by the enzyme nitrate reductase, passing down 2 electrons. For example, E. coli can catalyze that reaction. Nitrite can be released into the environment. and can be picked up by another organism. Nitrite can now, by a different enzyme, nitrite reductase, is going to be produced into nitric oxide. Nitric oxide is going to donate electrons by nitric oxide reductase and produce nitrous oxide. And nitrous oxide eventually, by the enzyme nitrous oxide reductase, is going to be formed into nitrogen gas. Those reactions from nitrate to nitrogen gas can be performed by denitrifiers like Pseudomonas tutzeri. The products of nitric oxide, nitrous oxide, and nitrogen gas, being gases, could be then released into the environment, and that will happen in a dissimilatory fashion. So here we have an image in between the respiration reactions and the nitrate-based anaerobic respiration. As you know, in a respiratory reaction, you're going to have, for example, an electron donor like NADH that is going to donate its electrons to a complex, and that complex is going to help pass protons across the membrane. The electrons will go into some kind of quinone pole that eventually is going to donate it to a set of enzymes that are going to reduce oxygen into water. All of those are going to generate protons that eventually will be used in the proton motile force to generate ATP. Now, when the cell uses nitrate to respire, you can have in this condition NADH donating the electrons. Those are going to result in protons being pumped across the bacterial membrane. Again, you're going to have a quinone pole that is going to also help pass those electrons. But at the end, what you're going to have is an enzyme, for example, the nitrate reductase complex, that are going to now take nitrate and accept those electrons to produce nitrite. And nitrite can now be released into the environment. So the denitrifiers, which are going to respire anaerobically to denitrification, will take the electrons again out of NADH, pass them through a complex that are going to pump protons across into the periplasm. They're going to also have a quinone pole system that is going to now bring those electrons to a nitrate reductase complex. And here you're going to have nitrate, which is going to accept those electrons and eventually become nitrite. As the electrons are passing by from this complex to the next complex, here is the cytochrome CD system that is receiving them. You're going to take these electrons by the enzyme nitrite reductase, and nitrite reductase is going to generate nitrous oxide. Electrons are going to be continuing down another set of electron acceptors and eventually get to the enzyme nitrous oxide reductase, which is going to take nitrous oxide plus protons and produce nitrous oxides. So nitrous oxide now is going to eventually donate those electrons and produce nitrogen gas. So as you see, it's a set of electron transport reactions like the ones that we have studied before, similarly in respiration in the mitochondria, that are going to eventually take the electrons from an electron donor and go down a pathway of electron acceptors to completely take nitrate and generate nitrogen gas. So the issue with denitrification is that it is a blessing and a curse. The bad news about it is that nitrogen gas, it's much less available to the organism than nitrite as a source of nitrogen. So you're basically losing it to the environment. So for agricultural purposes, denitrification is a detrimental process. In agricultural reactions, you are giving a fertilizer to increase the amount of nitrogen in the system. So if nitrate is used and converted into nitrogen gas, that is going to escape into the environment and not be used as a fertilizer for the plants that actually need it. So agriculturally speaking, it's very bad to lose nitrogen through denitrification. However, on the other hand, denitrification is very beneficial for sewage treatment, because in the water, when you have nitrate, those reactions are going to be able to remove the nitrate from the water system and make nitrogen gas. That nitrogen gas is going to be released. That is going to eventually help reduce the amount of algae present in the water treatment process. So the denitrification reactions together with the Anammox reactions that we learn are going to be able to remove ammonia, amines, and nitrate from the sewage system treatment water and release it to the environment in the form of nitrogen gas, and therefore eliminating the possibility or reducing the possibility of producing algal blooms in the water sewage treatment. So that is going to bring down the load of nitrogen in the water that can stimulate algal cell growth. One last reaction that I want to bring up to you are there going to be the ammonifiers. And ammonifiers are going to take nitrate and eventually bring it into ammonia. So now it's going to happen by two reactions. One of them is going to have the enzyme nitrate reductase, and the other type of nitrate reductase, different from the one in the nitrifiers, are going to eventually get the nitrite and make it into ammonia. Now, E. coli and many enteric bacteria in our gut are able to have these reactions to produce the ammonia and therefore bring that ammonia back into the biological system. Last but not least, I want to bring up to you the reactions of nitrogen fixation. These are the reactions which are going to be either anoxic or oxic and are going to able to bring the nitrogen gas from the environment back into the biological system as the form of ammonia. So take a look at this little plant that is growing in a very barren environment. In this kind of environment where this little plant is thriving, you do not have a lot of ammonia. Nitrogen is going to be one of the main limiting reactions of many plant-based lifestyles, and therefore giving nitrogen to a plant, as we do with fertilizers, is going to make it thrive and help in the production, for example, of food. So cells must acquire nitrogen for growth, and in this case it's going to be bacterial cells. They need to bring in nitrogen. So they can bring it in because they can either pick it up from amino acids, they can bring it by ammonia, or from nitrate. Now here's the reaction in which the nitrogen fixation is going to be used to bring that energy, but that nitrogen, back into the biological system. The problem with this reaction, shown over here, where you have a nitrogen gas, you're going to have to require a lot of electrons and protons to get two ammonia molecules and a hydrogen gas. It requires a lot of energy. It's an endergonic reaction. So it's going to have a positive delta G naught of 275 kilojoules per mole. So in a bacterial cell, you can have a transporter that is going to be able to bring ammonia in. You're going to have one that can bring nitrate in, and eventually that can be converted to ammonia, or amino acids could be transported by an ABC transporter, and those go directly in, and they contain also nitrogen. But nitrogen gas can diffuse freely across the membrane, and microorganisms that can fix it will be able to have the energy to make it into ammonia. Now, many bacteria species are able to fix nitrogen. Many are obligate aerobes. Some of them are going to be facultative aerobes, and some of them are going to be strict anaerobes. They are going to be growing in quote-unquote low nitrogen environments. So fresh water, ocean floors, soil, deserts, etc. Now, you have cyanobacteria, Clostridium microorganisms in soil, azobacteria, bacillus, rhizobium in relationship with plants, and Frankia in relationship with plants. All those microorganism species are able to fix nitrogen. Some archaea are also able to fix nitrogen, most of them anaerobically, and some of them, the methanogens, are able to also fix nitrogen to bring it into their biological systems. So this table here shows some of the nitrogen fixing organisms. Some of them are going to be chemoorganothrobs, like azobacters. Other ones are going to be phototropes, like cyanobacteria. And some of them are going to be chemolithotropes, like alkalogens. Those three examples that I gave you are from free-living aerobes. You also have free-living anaerobes, Clostridium microorganisms in the chemoorganothrobs. You have some of the purple photosynthetic bacteria like Chromatrum or Rhodobacter. And you have some of the chemolithotrophic bacteria like Methanococcus. Those are the examples of the free-living organisms able to fix nitrogen. What I want to also remind you is that there are going to be some microorganisms that can do this symbiotically, like the rhizobium with the legumes, or the Frankia with the alder, bayberry, or the autumn olive trees. So some of the bushy plants that we can find. Now, this reaction, it's going to be catalyzed by one of the mostly studied enzymes for nitrogen fixation, and that enzyme, it's called nitrogenase. Nitrogenase enzyme, you have seen part of it before because I show you one of the many complex molecules, and I'm going to show it to you again today. It's widely distributed enzyme in many bacteria and archaea, so it's very conserved, and it has two main components. You have a denitrogenase component that has iron-sulfur complexes, and then you have the dinitrogenase reductase, and that has the iron-sulfur molybdenum coenzyme component. This enzyme, however, is very sensitive to oxygen, so the microorganisms have evolved mechanisms to prevent the amount of oxygen present anywhere close to the nitrogenase enzyme, so oxygen cannot inhibit the reaction. Some of them are going to secrete slime layers around themselves to reduce the amount of oxygen diffusion. Other ones are going to use oxygen by respiration at really high rate, so every time that oxygen comes in, it's eventually going to be consumed immediately. And some of them are going to have compartmentalization. They're going to have different structures within the microorganism in order to have those reactions. Other ones, like the microorganisms of the cyanobacteria, some of the cells will develop into special cells that can only do nitrogen fixation. So in this case, let's take a look about the reaction that is going to happen and some of the energetic requirements that this reaction is going to have. So what we're going to look in here is that by whatever means, either photons, NADH, hydrogen, or pyruvate, those molecules are going to pass their electrons to a reduced ferredoxin. And the reduced ferredoxin eventually are going to pass the electrons to the nitrogenase reductase. That's the one containing the iron-sulfur system. Notice here the large amount of ATP that it requires. It's going to use the energy of 16 ATPs in order to catalyze those reactions. Eventually, those electrons are going to pass to the nitrogenase enzyme portion that has the iron, molybdenum, and sulfur complexes. That part of the reaction, using 8 electrons, it's going to now catalyze the fixation of nitrogen, where nitrogen will be taken in and produce 2 ammonia molecules. Here, to remind you, it's an example of the iron, sulfur, molybdenum coenzyme that is part of the nitrogenase enzyme. So here, this is the part that is actually going to work to reduce nitrogen gas. We have 2 of these molecules per enzyme, and depending on the enzyme, this little molybdenum atom that is present in here could be changed for either an iron or a vanadium, or sometimes iron alone, depending on the enzyme, especially when molybdenum is scarce. We talked about molybdenum being one of those trace elements that is require very low concentration. So if the cell, doesn't have it, it will be able to substitute another molecule like a vanadium or iron into that position to continue allowing it to fix nitrogen. Interestingly, the nitrogenase enzyme in archaea only have iron in that position. So this chart over here, this flow chart, it's going to show you the flow of electrons catalyzing the reaction. This reaction, it's going to begin from pyruvate, and eventually it's going to have the electrons being accepted by nitrogen gas. The way that the reaction is set up, the molecule that is bolded, it's the one that is having the electrons. So what we have over here at the beginning, it's a pyruvate molecule, so 4 pyruvate molecules by the enzyme pyruvate flavotoxin oxidoreductase. It's going to take 4 pyruvate and 4 acetyl-CoA molecules and remove 4 times 2 electrons to generate 4 acetyl-CoA and 4 carbon dioxides. So the pyruvate is going to donate the electrons to the flavotoxin. The flavotoxin is going to be the other partner, 4 of them, which are going to be reduced, shown over here by this bold flavotoxin word, and contain the electrons now. Now the flavotoxin now is going to be able to pass those electrons to the dinitrogenase reductase enzyme, which is going to, as you see over here, go from the oxidase state to the reduced state. So now we have 4 dinitrogenase reductase enzymes that are being reduced. The electron transfer now is going to happen, and the electron transfer to dinitrogenase is going to be happening one at a time, and it requires 2 ATP molecules per electron to catalyze the reaction. When we have 4 dinitrogenase reductase enzymes passing its electrons to dinitrogenase, it's going to require 16 ATPs. Eventually, you're going to have the dinitrogenase enzyme being reduced, and now the electrons that are being harbored by dinitrogenase are going to be passed to nitrogen gas to create 2 ammonia molecules. So at the end, if you look at the nitrogen molecule, you're going to also have the production of hydrogen gas, which is happening somewhere else in here. You eventually are going to have the complete reduction of nitrogen gas into ammonia by a set of consequent reactions shown over here at the bottom, taking 16 different ATPs to generate the energy. One way that we can detect the presence of that enzyme complex in bacteria is to have the catalyzed reaction of acetylene to ethylene. So acetylene, as you can see here, it's a molecule of 2 carbons and 2 hydrogens. You have 3 bonds between the acetylene. And when you have protons and a dinitrogenase enzyme, you can now produce ethylene, which has now only 2 double bonds. Excuse me, I only have 1 double bond. So you have gone from a 3-bonded carbon to a 2-bonded carbon. We can use that substrate, acetylene, to detect the presence of the hydrogenase enzyme. So to conclude, what we're going to look is that there's a lot of nitrogen gas being fixed on Earth. We consider that it's 280 million tons per year. And a lot of that is happening biologically. About 85% of that nitrogen fixation is biologically. Some of them can happen during fertilized plants or with lightning, actually. Microorganisms, in particular prokaryotes, are the only ones who can fix nitrogen, and therefore we need them to bring nitrogen gas from the atmosphere back into the biological system. Plants and animals therefore form symbiotic relationships, for example, with bacteria, so they can take that nitrogen and bring it into ammonia, nitrate, or organic nitrogen. Another part of nitrogen fixation is that it's mostly occurring inland. About 60% of it is occurring inland, mostly by free-living bacteria, but also by symbiotic bacteria in the roots of plants. So a plant-bacterial symbiosis, for example, we consider that contributes between 55 and 165 pounds of fixed nitrogen per square mile per year in some types of soil. And 40% of it is going to happen in water, mostly by free-living bacteria. With that, I'll stop. Eventually want to show you this image where you can have a soybean plant on the left without the bacteria which symbiotically can fix nitrogen, and the one on the right which have a symbiotic relationship with a bacterium, Rhizobium, that is able then to bring nitrogen gas into the system, and therefore you can appreciate that the microorganisms growing with the symbiotic relationship with the plant are able to contribute to the well-being of the plant. The plant looks a lot healthier, has grown larger, is a lot greener than the plants on the left, which do not have the microorganisms to help them acquire nitrogen. I'll see you then on Tuesday.